Hello students, how are you all? I hope you all are doing well. I am Mr. Prithviraj. As you all know, I am your 9th class biology teacher. So today we are discussing your 4th chapter, the most important one, that is your plasma membrane, or you can call it cell membrane. Plasma or cell membrane. So before I start my class, I would like to tell you second wave of corona is coming. So please take care of yourselves. Okay. And read well. In our last class, we have read about our cell organelles, the important cell organelles, their functions, a lot of stuff we have discussed in our previous classes. Okay. In this particular chapter, we will specially discuss about cell membrane, means plasma membrane, how important it is. Okay. So in this lesson, what all things we will cover that I need to tell you. Today we will discuss how can we create solution. First topic today I tell you, that is how to prepare solution. Okay, we will discuss how to prepare solution. In that I tell you two kind of things. First, we will know how to prepare sugar solution. It's easy, but we need to understand in a scientific manner. Okay, how to prepare preparation of sugar solution is our first topic. Second topic is preparation of concentrated sugar solution. These are the two topics which we will discuss, which we will cover in this class, and later we need to know an important activity. Okay, that is your what? Lab activity number 1. So before we start those, let us complete with preparation of solution and how to prepare a concentrated sugar solution. First thing, how to prepare a sugar solution. In your book, it's your first activity, I think. It's your first and most important one. Okay, so let us understand how you will make it. Situation number 1. What we will take? We will take one beaker. Consider this as a beaker. Now what we will do? We will add two spoons of sugar. What we will do, students? We will add 2 spoons of sugar in what? 100 ml of water. This is our 100 ml of water. In this 100 ml of water, we will add 2 spoons of sugar. After adding, you will see it will be dissolved. Right? So in this case, you will call water as solvent. And you will call sugar as solute. Solvent plus solute will make a solution. This is what you need to understand from this particular topic. This is your first one. Second phase is how to make a concentrated sugar solution. How will you make? Instead of taking 2 spoons, let us take 3 spoons. How many spoons we will take? Let us take 3 spoons. When we will take 3 spoons of sugar, what will happen? That sugar will be dissolved in water and little amount of sugar will be left undissolved. You will find little amount of sugar that's left undissolved. No matter how much you try, you will see that sugar is not dissolved in that 100 ml of water. That means now the water has lost its capacity to get or to accept more sugar solutes in it. So here in this case, this particular solution is said to be a concentrated sugar solution. To be frank, whatever we have discussed, that this is a solute, this is a solvent, solute is your sugar and solvent is your water. Okay, so this is quite same, but only thing is you cannot add more sugar in it. It's finished, it's full. Okay, now what is concentrated solution? Now we will understand what is concentrated solution. That is your second most important topic in your book. It is given, so I'll make it a little bit easy for you guys. Another topic is here I need to cover. Concentrated solution, that is short form for solution. I wrote SOL to the power n, that is your short form for solution. Now you will see what is concentrated solution. To conduct this, what you need? You need 3 particular beakers. How many beakers we need? We need 3 beakers. Let us do this. Add 100 ml of water to it. In all these 3 cases, what we will do? We will add 100 ml of water. I hope you all can see. These are 100 ml of water which we have added to all of them. Now what we need to do? We will add sugar. How we will add? We will add here half spoon of sugar. How much? Half spoon of sugar to beaker number 1. Okay, then we will add 1 spoon of sugar. 1 spoon of sugar to beaker number 2. Now beaker number 2 got 1 spoon of sugar. Now beaker number 3, this will get 1.5 spoon of sugar. How much? 1.5 spoon of sugar. Now you guys need to answer me. Which is the most concentrated one? Obviously this, because the concentration of sugar is more in which case? Third beaker as compared to second and first one. And which is having least concentration of sugar? First one is having least concentration of sugar. That is also very clear, right? Now the amount of sugar present in 100 ml of water is called concentration of sugar in that particular solution. We will tell that amount of sugar present in 100 ml of sugar, 100 ml of water is called concentrated solution. Whenever somebody asks what is concentrated solution, you need to say the maximum amount of solute, the maximum amount of solute can be added to 100 ml of solvent. Maximum amount of solute can be added to 100 ml of solvent will be the condition for making a concentrated solution. Is that clear? I hope it is clear. Now let us conduct our lab activity. Most important lab activity. So I hope you all will pay attention here. Now, lab activity number 1. Why we are conducting this lab activity? This particular lab activity will help us to understand osmosis. Okay? This particular lab activity will make us to understand what is osmosis, how exactly it is, how can we see live example of osmosis. All this stuff we will be getting from this particular example. Please pay attention. So let me write here. Whenever we are doing a lab activity, following process need to be conducted. First thing you will write is the aim of the experiment. What exactly is the aim of this particular experiment? If an exam comes, you need to write in this format. I have already told in your previous classes also. First you will write aim of the experiment. Aim of the experiment is to study the process of osmosis with the help of dried resins in different solutions. What? To study the process of osmosis with the help of dried resins. Okay, where in different solution. In different solutions. Is it clear? Our aim is to study the process of osmosis in different solutions with the help of resins. You know kismis, right? Yes, that's an example of resin. Now, what are the materials you need to require? That is very important. You need to write here. What you will write? Materials. What are materials we need? First material we need is 2 beakers. How many beakers we need? We need 2 beakers. Then we need tap water. Yes, we need tap water. Little bit of tap water. Then we need sugar. Okay. We need dried resins. We need dried resins also. Then what we need? And we need, or you can, that's it. You can take dried raisins also, or you can take dried grapes also. That's it. This is the only thing which we need. Now, the required materials also we have got. After this, you can do activity. What you will do? First, you will do primary activity. First of all, we will conduct the activity, then we will see what exactly is happening. Okay, so let us understand how this activity goes on. We will take a beaker. What we will do? We will take a beaker. Okay, now in this beaker, we will put 100 ml of water. How much amount of water? 100 ml of water. Which water we took? Tap water we took. 100 ml of water is also there. In this 100 ml of water, we will place kismis, or you can tell dried raisins. Okay, we place 2 dried loaf of raisins over here. We need to leave it overnight. Okay, then observation. This is our primary activity. So in this activity, what we have observed, in the next step you need to write primary observation. You will write primary observation. In the primary observation column, see aim, materials, primary activity, then primary observation. In primary observation, we have seen that what we have seen, the raisins have swollen. The raisins have swollen by absorbing water. You will write that only. By absorbing water. This is what our primary observation is. Now, what we will do? Now we will see again another experiments. In this only, second activity we will do. What is final activity? We will take 100 ml of saturated sugar solution. Okay, let me erase this. See, primary activity done. Now we will do final activity. After that, what I write? I write here final activity. And we will see final observation also. Then we will see what is our final observation also. So in our final activity, what we will do? Again we will take one beaker. In this beaker, instead of taking water, we will take 100 ml of concentrated sugar solution. Is where no more sugar can be added. Now in this 100 ml, we will place again swollen raisins. In our primary observation, we have taken 2 swollen raisins. Now I will put those swollen raisins over here. Now I leave it overnight. Now what I see, those swollen raisins again they are dry. Okay, what will happen? Swollen raisins lose water. Swollen raisins will lose water and they will shrink. So what exactly we have learned? After writing all this, aim, materials, primary activity, primary observation, final activity, final observation, what are things we are getting from this? Now you need to write learning. In learning, what we have learned, the weight of the swollen raisins is more than that of dry raisins. This is because the cells present in the outer layer of the raisins permit water to pass through it. First thing is, in the first observation, what we have seen, those were dried raisins. After keeping inside tap water, they have swollen. Okay, so the weight of the swollen raisins are more than that of dried raisins. Why? Because the outer layer of the raisins accepted water. They allowed the water to pass through it. When water passed through it, now it has been swollen. Again, when you are putting inside your concentrated solution, you see they lose their weight. Why they lost their weight? What exactly is the reason why they lost their weight? This particular thing we will discuss in our next particular observation. Until then, bye bye.